আমরা ব্লাড গ্রুপিংয়ের আলোচনা এর আগে করেছি, কিন্তু নোটসটা আমি একটু পরপর বলে দিচ্ছি, কারণ যদি কোনো লেখা পড়তে না পারো তাহলে বুঝতে সুবিধা হবে। First, introduction. Blood in different individuals differ immunologically according to the antigens present in their RBCs, and according to this antigen, blood or individual form different groups called blood groups. Definition of blood group. The term blood type or blood group refers to the type of antigen present on RBC membranes. There are two main systems, example ABO system and RH system. ABO system. In blood group, two terms are very important. Those are agglutinogen or antigen and agglutinin or antibody. Agglutinogen. What is agglutinogen? The surface of the red blood cells carries a range of different proteins are called agglutinogen or antigen. These antigen are complex carbohydrates, mainly glycosphingolipids and glycoprotein in nature. Example, antigen A, antigen B and antigen H. Antigen H is the precursor of both antigen A and B. Agglutinin. What is agglutinin? The term agglutinin is used to describe antibodies dissolved in plasma that react with specific blood group antigen and agglutinogen. Example, anti-A antibody and anti-B antibody. Next, antigen and antibody present in four blood group: AB, ABO blood group, and antigen on RBC's membrane. In blood group A antigen present and anti-B antibody in plasma. In B blood group, B antigen is present on RBC membrane and anti-A antibody in plasma. In blood group AB, A and B both antigen are present on RBC membrane and no antibody present in plasma. In O blood group, A and B both antigen is not present and anti-A and anti-B antibody are present in plasma. Antibodies are of IgM type, IgG also, and are produced in the body in sufficient amount after birth, may be due to exposure to the respective antigens through food, intestinal bacteria, etc. Landsteiner law. If a person has antigen A, he will not have anti-A or alpha agglutinin. If one does not have an antigen, he will have its antibody. Example, group A will have anti-A antibody. Example, group A will not have anti-A antibody but will have anti-B antibody. Importance of ABO system. ABO system is very important for blood transfusion. Mismatch may lead to death. Cross-matching should be done before blood transfusion. Cross-matching is done by using ABO system and RH system. Every group have some blood group related disease. Example, secretors of O group may suffer from peptic ulceration. Study of blood group are of great importance of anthropological survey. ABO system and blood grouping. is very useful in pinpointing an individual and sometimes becomes of great help in criminal investigators. In an incompatible blood transfusion, antibodies in recipient's plasma bind to the antigen on the donated RBCs, which causes agglutination, that is clamping, of the RBCs. The complement molecules of our defense system causes hemolysis or rupture of the agglutinated RBCs, and the released hemoglobin may cause kidney damage, clogging of filtration membrane. Universal donor. As O blood group people can donate, all blood group people AB, O blood group is known as universal donor. Universal recipient AB blood group people can receive blood from all blood group people AB, AB blood group is known as universal recipient. The Rh blood group is so named because this antigen was first discovered in the blood of rhesus monkey by Landsteiner and Wiener on 1940. Rh means rhesus. Rh positive blood means that Rh antigen or D antigen is present on its RBCs. 85% of people have this antigen, and Rh negative blood means that Rh antigen or D antigen is not present. 15% of people have no antigen, so they are Rh negative. Anti-Rh antibody. The antibody in Rh system is called anti-D antibody. Is produced only when an Rh negative individual receives the D antigen. The antibodies are of IgG variety. These are small enough to cross the placental barrier. Importance of Rh system. The Rh system in relation to transfusion leads to long-term complications. Example, if Rh negative individual is transfused with Rh positive blood, then in the recipient anti-D antibody is produced. These antibodies remains in the recipient blood. Second transfusion of Rh positive blood into the bloodstream of same person will cause agglutination and hemolysis of the RBCs in donated blood occur. If a Rh negative woman conceives Rh positive baby, if the husband being Rh positive, then during childbirth, some fetal RBCs with D antigen can pass to the mother. In the mother, they produce anti-D antibodies. In the next pregnancy, if there is Rh positive fetus, which is usual, these antibodies from the mother will lead to destruction of the fetal RBCs, leading to hemolytic disease of newborn. Short form is HDN, hemolytic disease of newborn. Rh positive husband, Rh negative wife. If first baby is Rh positive after childbirth, some fetal RBCs with D antigen can pass to mother. Next, in the mother, they produce anti-D antibodies, and next pregnancy, if fetus is Rh positive, antibody from mother will lead to destruction of fetal RBCs, leading to hemolytic disease of newborn. In mild degree of destruction of fetal red cells, it leads to anemia and rapid RBC production. Therefore, blast cells, erythroblastosis, come into circulation, and this is called erythroblastosis fetalis. It is also associated with jaundice. In severe form of fetal RBC destruction, more instance jaundice occur, icterus gravis neonatorum. Treatment of hemolytic disease of neonates. It can be prevented if the mother, Rh negative mother, is injected with anti-D antibodies immediately within 72 hours of the childbirth. These antibodies then destroy the Rh positive RBCs of the fetus, which may have entered into the maternal circulation. This prevents development of anti-D antibodies in mother. Coombs test. In hemolytic disease of newborn, the Rh antibodies from mother's blood pass through the placenta and combine with the Rh antigen on the fetal RBCs. Such RBCs are called sensitized RBC. Coombs test is used to detect these sensitized RBCs and also to detect Rh antibodies in maternal blood. Cross-matching. Cross-matching is necessary to avoid reaction due to unknown antigen and antibodies. Process. In cross-matching, the recipient's RBCs are treated with donor serum and donor's RBCs with recipient serum. If no agglutination occurs, only then transfusion is given. Test of donor's RBC with recipient's blood is more important than donor serum, and donor serum is not important as it gets diluted in recipient's blood. Importance of cross matching. The knowledge of cross matching is necessary for blood transfusion mainly. If a mother of Rh negative blood conceives Rh positive baby, husband being Rh positive and mother is already having anti-D antibodies due to previous transfusion of Rh positive blood, then the first baby will be affected with hemolytic disease of newborn due to passing mother's anti-D antibody to fetus blood circulation. Donor. Donor is the person who donates blood for transfusion to other individual. Recipient. Recipient is the person who gets blood for transfusion from donor. Group O Rh negative blood group can be named as universal donor, and AB positive blood group can be named as universal recipient.